Hello, my name is Katherine Do, and welcome to the Module 1 Database Generation video for the Clinical Metaproteomics tutorial. The video preceding this video provided an overview of this tutorial where we covered the background of the metaproteomics field and why we developed this workflow. If you haven't already, please watch that video in order to gain better context for this video. Aside from the overview video, there is a total of 5 modules. In this video, we will cover the first module, Database Generation. The agenda for this video includes a recap of the Clinical Metaproteomics tutorial, as well as how to set up for a module, followed by the main focus of this video, executing the database generation workflow. In the previous video, I gave an overview of the Clinical Metaproteomics tutorial, but I will give a quick refresher here. As I mentioned, this video covers the first module, Database Generation. In this video, we will obtain a comprehensive protein database that includes proteins from humans, contaminants, and our various species of interest. Here we generated our own comprehensive database by performing a literature survey because we didn't have a reference database or information from 16S rRNA sequencing data. Because the comprehensive database contains millions of sequences, we will use the MetaNovel tool to reduce the size of the database. This reduced database will be used for the discovery module. In the discovery module, we will use 2 peptide identification programs, SearchGUI Peptide Shaker and MaxQuant, to identify peptide and protein sequences via sequence database searching. This will allow us to retrieve reliable microbial peptides. Next is the verification module. We will use PepQuery2 to verify confident identification of microbial peptides to avoid misassignment of human peptides. We will then create a database of protein sequences associated with these verified peptides. This verified database will help us reduce false positives when we quantify our proteins and peptides. In the quantification module, we will use MaxQuant to quantify microbial and human peptides and proteins separately to compare results. Lastly, we have our data interpretation module. Here we will perform statistical analysis using MS-Stats TMT and taxonomic and functional annotation using Unipept. Next is the hands-on section. For this hands-on section, we will go on the Galaxy EU website to set up a new history, import data, in the workflow, and start the workflow. In the previous video, I mentioned that we will use the same setup for all 5 modules. In the interest of time, we will start the database generation workflow first, and then talk through what it does. Now let's go to the Galaxy EU website. In the previous video, I mentioned how a history on Galaxy is similar to a search history on your web browser. We will make a new Galaxy history for each module so we can understand where each module begins and ends. The Galaxy EU website is laid out to be user-friendly. On the left panel, you can import data and select tools to use. The top bar lets you access workflows and data. The right panel displays the history we are currently working with. This center area will display your current task. If you select a tool or want to view a dataset, this is where you can see it. To make a new history, click on the plus icon on the right-hand panel. Then click on the pencil icon to name your history something appropriate. For this module, I will name it CMP for Clinical Metaproteomics, the number 1 for Module 1, _ the name of the module, which is Database Generation. Then I will click Save. If you need a quick refresher in the future, you can check out the FAQs on the GTN website. Now I will show you how to import data in the workflow. The FAQs cover these steps as well. I will show you how to import data through links. This method is also shown in this yellow tip box on the GTN tutorial page. While on the GTN tutorial page, go to the section called Data Upload. You can copy all the links in this black box by clicking on the icon in the top right corner. Then switch to the Galaxy EU page where you have your history that you just made. On the left-hand panel, click Upload Data, and then Fetch Paste Data. Paste the links that you just copied in the text field, and then click Start. You will see the datasets appear in your history. When everything is done uploading, click Close. Now, looking at the inputs, there are 4 MGF files and 3 FASTA files. The 4 MGF files are our example sample data. To keep things organized, we will create a dataset collection that will allow us to select all of our sample data as one input, rather than select each dataset individually. We will do this for later modules as well. To create a dataset collection, first click on the check mark icon on the History panel. This will allow you to select the items that you want in your collection. Click on the button that says how many datasets you've collected, and then build Dataset List. Type a name, such as MGF Collection, and click Create List. Finally, click on the checkmark icon again to deselect the datasets. You have successfully imported all of your input data for this module. Next, we want to get the workflow. We've included the workflow link on this slide and on the GTN tutorial page as well. You can open that workflow link in your web browser and it will take you to the workflow preview. If you remember, I mentioned in the last video that a workflow link contains a series of tools that have been preset and is ready to run by itself once you give the inputs and start it. For this workflow, click on the import link and we will start using this workflow now. If you look at the top bar, we are now in the workflow section. To start running this workflow, click on the play button and we will start putting in our inputs. Now we will start putting in our data. In the interest of time, I will start the workflow first and then explain the input data and workflow steps. So now we've imported our workflow. We've set up the input names, so we just need to match the datasets as shown. Once you are done, click Run. Now let's cover the inputs. We have provided 4 example datasets as MGF files. Previously, we put these in a dataset collection in our history to keep things organized. These datasets were originally generated as raw files, but for software compatibility, we have converted them into MGF. Later modules are able to use raw files. These data originated from Pap test fluid or PTF samples that were collected from ovarian cancer and non-ovarian cancer patients. We thank Dr. Amy Skubitz and her lab at the University of Minnesota for allowing us to use this data for this demonstration. I will summarize the sample processing for these datasets. Proteins from each sample were isolated, digested into peptides with trypsin, and labeled with a distinct tandem mass tag, or TMT, 11-plex tagging reagent. The TMT tagging reagent will aid us in quantifying proteins. Next, samples were pooled and separated by reversed phase liquid chromatography and analyzed by liquid chromatography tandem mass spectrometry, or LC-MS-MS, to generate MS-MS datasets. To save you time, we have also provided 3 protein sequence databases. All of them are in FASTA format, which is a text-based format for storing nucleotide or amino acid sequences. To see details on how we generated these, please see the tutorial on the GTN website. The Species Protein Sequences database accounts for 118 taxonomic species of organisms that are commonly associated with the female reproductive tract. We obtain these species from looking through literature. These are the microorganisms that we are interested in. If we find any matching sequences in our sample data, we want to retain these. We also want to include human and contaminant proteins in the database because these samples came from humans. Any matches to human or contaminant proteins in our sample data will need to be filtered out. Here is the overall process for the database generation module. I will give a summary of the module here, and for the next couple of slides, I will talk about the intermediate outputs and end with the final expected output for this module. For specific tool options and parameters, you can refer to the written tutorial on the GTN website. So, in the top right corner, this is where most people start. This is when we just have sample data that we want to analyze with a bioinformatics workflow. In this module, we have provided 4 datasets that have been analyzed by LC-MS-MS, and we have it in the form of mass spec datasets. We will also need protein sequence databases to search the sample mass spec data against. The 3 databases that we mentioned on the last slide will form our large comprehensive database, which will cover what we are and are not interested in. The Species UniPro database contains protein sequences for the microorganisms that we expect to see present. Then we have the Human SwissProt database. This contains human proteins since these samples originated from humans. Lastly, we have the Contaminants, or CRAP, database that accounts for potential dust and contact proteins that one might find in a lab or when handling samples. We will merge these 3 into one large comprehensive database that will consist of millions of sequences, which many tools cannot handle. So we will perform database reduction using the Metaproteomics tool. This much smaller database is easier to manage. We will merge this small database with human and contaminant protein sequences for a database that we will use in peptide discovery. As you can see, even after merging, we will have a database that is around 21,000 sequences, which is much more manageable than millions of sequences. Now let's talk about the workflow in more detail. First up is the large comprehensive database for Metaproteomics. We provided 3 separate protein sequence databases, and we will merge and filter these sequences to obtain a large comprehensive database for Metaproteomics. As you can see, the number of total sequences will have gone down because we have filtered out duplicates. However, the size of this database is still in the millions of sequences, which many tools cannot handle. Therefore, we need to reduce the database size. For that database reduction, we will use the Metaproteomics tool. So what does Metaproteomics do? The Metaproteomics tool infers proteins and organisms directly from raw mass spec data in the input protein sequence database to generate a reduced, targeted database. The compact protein sequence database can be searched against raw mass spec data with improved sensitivity and search speed, while also reducing the number of potential false positives. This brings us to the last step for this module. The compact Metaproteomics database will be merged and filtered with the Human SwissProt and contaminant databases to generate the reduced database that will be used for peptide discovery in the next module. We perform this merging and filtering step again because we want to make sure that any human and contaminant matches can be selected out later on in our analysis. If you cannot generate this output, that's okay. We will provide you with the inputs for each module. This marks the end of Module 1 database generation of the Clinical Metaproteomics tutorial. To navigate to the next tutorial, go to the bottom of the database generation tutorial page until you see the Go Further box. Here you can click on the link for the next tutorial. We hope that this was interesting and hope to see you in the next video where we will cover Module 2 Discovery.